Hi, so today we will be going over chapter 10 of Kaplan, homeostasis. We will be beginning with the kidney. So the kidney's main function is to excrete waste. We'll talk about much more secondary roles in the later part. So we have two kidneys, bean-shaped structures towards the neurodominum located in the back. And for the MCAT, you really have to focus on the specific functionings of the cell. So we have what's called a nephron, Nephron, in our kidney. And really, it's important to see that we have kind of like this cortex and outer cortex with an inner medulla. Outer cortex and an inner medulla. And this type of structure really shows up all over the body. In our brains, we have the cerebral cortex with the medulla oblongata. In our adrenal glands, we have the cortex, which makes many steroids, and we have the medulla, which makes catecholamines. So this type of structure is prevalent everywhere. And in terms of the MCAT, you really have to know how a neuron functions, the different components, nephron functions, the different components of a nephron, which we will go over now. So, we have circulatory, so our afferent artery comes in and our efferent artery leaves. So we know one of the biggest functions is that it's going to filter the blood, and the site of where this happens is referred to as the glomerulus, and sometimes it's located within the Bowman's capsule. Bowman's capsule. And just know it is the site of filtration. So filtration is pretty much, you allow some things to go through and some things you don't allow to go through. So some of the things that do go through are stuff like, let me just draw this tubule. So things that do go through are stuff like, salts, NaCl, you have glucose, vitamins, amino acids. The things that do not go through are proteins and red blood cells. So erythrocytes, they aren't going to go through. So that's important to know, because if they do go through, that means you are having kidney, malfunction or kidney failure. So we do not want proteins or red blood cells to go through. So, the blood comes in from here, and it gets filtered, and it goes out in the efferent arteriole. Now, we move on to this next piece right here, which is referred to as a proximal convoluted tubule. So I'll just write that here. Convoluted tubule. Now, this is where all the stuff that was filtered in is going to pass through. So there's two important functions or roles that occur in the proximal convoluted tubule. And so the first role is what's called absorption, or specifically reabsorption. And what that means is all the stuff that was filtered out, like salts, glucose, amino acids, our body does need them in order to function. So we're actually going to reabsorb them into our circulatory system. So there's going to be a vessel that's near the proximal convoluted tubule. And so the flow of solutes is going to go from the proximal tubule into the vessel. I believe it's specifically the vasa recta. But this is just a process of reabsorption. We are taking stuff from the kidney and putting it back into our body. Reabsorption. So we're going to reabsorb things like salts, glucose, vitamins, amino acids. Next, another thing that occurs is secretion. Should have done that in another color, but secretion is pretty much, we're now putting, instead of like, reabsorption was we're putting stuff back into our body. Secretion is we're putting it into the kidney, which will eventually go out of the body. So we're pretty much putting stuff out of the body. So things that we want to eliminate, such as ammonia, like urea, or potassium, or hydrogen. So secretion is when we're going from the body into the proximal tubule. And the main one you should probably know is urea, which is just a nitrogenous waste. So we had Bowman's capsule, which was filtration, the proximal convoluted tubule, which is areas of reabsorption and secretion. And next we move on to a loop of Henle. Just move this right here. So there's a couple things that you have to know about the loop of Henle. You do have to break it down into two parts, the descending and the ascending. So what's going down like here, this is the descending, and what goes up is the ascending. So what you do have to know is that descending is permeable to water. So water will actually leave, H2O. We'll get into why this is, because typically you move solutes and then the water follows due to osmotic pressure, but for the kidney it's a little different, and we'll talk about why. It's specifically due to the counter current multiplier. So water comes out here, and on the ascending it's permeable to solutes, things like NaCl-. Okay. So now let's talk about what that means in terms of molarity, or specifically osmolarity. So if we have, I'll just say A is here, B is here, and then C is right here. If I were to ask you, which one has the highest osmolarity concentration, would it be A, B, or C? The answer would be A. Now let's talk about, or sorry, my apologies, the answer would be B. This would have the highest osmolarity. So let's talk about why. So at A, we'll say we have a specific moles over liter, molarity. So moles over liter. But now descending, you're losing water, so you're losing volume. So your liter goes down. And if you know the bottom, if the bottom part of the fraction goes down, your osmolarity will go up. So B will have a higher osmolarity than A. Now, C, after, so we'll just put B has a specific molarity. And now we know the ascending is permeable to solutes. So what that means is the solutes are going to leave, so the molar value is going to decrease. So B will start decreasing until it gets to C. So C is going to be less than B. So B is going to be the highest osmolarity. And typically, if you go back to when we were talking about the cortex versus medulla, this is a prime example of, there's technically like a line that's going to be dividing the cortex and the medulla. So it's technically the lower part of the Henle, the loop of Henle, dips into the medulla. So typically the medulla of the kidney has the highest osmolarity concentration. Okay, so moving on, we have what's referred to as the distal convoluted tubule. This is the distal convoluted tubule. Oh, spelled that wrong, convoluted tubule. Okay, so same thing as a proximal convoluted tubule, we're going to get secretions, there's going to be reabsorption, but one of the main things that you should know about the distal convoluted tubule is that is where aldosterone is going to come and act. So what does aldosterone do? Well, what it does is, let's suppose we have the circulatory system, so we have a vessel right near the distal convoluted tubule. Aldosterone will be released, it will come and bind to a receptor in the distal convoluted tubule. We'll say it's this green right here, we'll just call that aldosterone. When it comes and it binds, it's going to exchange sodium for potassium. So we're going to put a lot of sodiums into the circulatory system. And as you know, if you move a solute somewhere, you're going to increase that osmotic pressure. So as we put sodium from the distal convoluted tubule into our circulatory system, we're going to increase the osmotic pressure. OP, osmotic pressure. And when we increase the osmotic pressure, fluid follows. So fluid will follow the solutes. So we are going to get a net influx of water, or H2O, from the kidney into our circulatory system. So what aldosterone does is that it retains water. So you know, things like when you're dehydrating or you know you're really thirsty, this is your body's going to act in effort to retain water. Now this very last, I'm just going to draw it again. So just continuing, so this was our distal convoluted tubule. It continues down to our collecting duct, where other nephrons will come and dump their content in also. So this is referred to as the collecting duct. Okay. And just like aldosterone, there is going to be another hormone that comes and binds in the collecting duct in order to retain water. And that hormone will be called Antidiuretic Hormone, or ADH, also known as vasopressin. This is going to be released from vasopressin. This is going to be released from the posterior pituitary gland. Okay. And it kind of has a similar function in that you're going to move water from the kidney to our circulatory system. So it works in a way to retain water also, but it works in a different way. It actually promotes aquaporins, or water channels, to just move water in. So if you have a circulatory system right here, it's just going to take a bunch of water and just move it in. So notice that the functionings are the same, as in, you know, they retain water, but the method is different. How is it different? Well, let's suppose we talk about aldosterone. We know that it moved a solute, so it moved sodium, Na, and then it moved water. So we get an increase in, so if we have moles, we would increase both moles and the liter. So pretty much that would cancel off, and the osmolarity of the blood would remain the same, because we're not only changing the moles, but we're also changing the liters. But since they're in fractions, they would cancel off. So the moles, the molarity of our blood would remain the same. However, this is not the case when we talk about ADH, or vasopressin. We are only moving water into our circulatory system. So we would increase the liter. In that case, since this is we're talking about the circulatory system molarity, we would actually end up decreasing molarity. So vasopressin, or ADH, is also used to control blood osmolarity. So when blood gets way too high, it can you know, release ADH, act on the collecting duct, and retain water. Okay, and eventually the collecting ducts, they all meet up, form the ureter, goes to our urinary bladder, and we can just eliminate it through our urethra. So now let's talk about some secondary roles that the nephron has. So I guess a better picture would be this one right here. This should be in the Kaplan book. This is pretty much everything that I explained. We start from the Bowman's capsule, filtration, then we move on to the proximal convoluted tubule, where you have both reabsorption and secretion. We move on to the descending, which is permeable only to water. And notice that we're in the inner medulla, so we're in the medulla, and this is going to be the highest level of osmolarity. Then we move up, which is soluble to solutes. We go on to here, the distal convoluted tubule, aldosterone will act on there. Then we have the collecting duct, where antidiuretic hormone would act on. Okay, so now let's focus on some secondary roles of the kidney. One of the biggest things that you should know that it also controls blood pressure. So how would the kidney control blood pressure? Well, there's a couple key factors that you have to know. One thing is that it has a group of cells known as the juxtaglomerular cells. And what these cells do is, for example, if the kidney senses low blood pressure, these cells will release an enzyme. Well, it's technically an enzyme hormone. So if you release something, it's called a hormone, but it functions as an enzyme. That's going to be called renin. And your liver is going to be secreting what's called... So liver is pretty much that's a pretty bad drawing of a liver, but let's say that's a liver. So your liver is going to be the source of creating blood proteins. So many proteins that are found in the blood typically come from the liver. So your liver is going to secrete what's called angiotensinogen. And so angio typically refers to blood vessels. Tensin, we're talking about blood pressure. And ogen is zymogens, are pretty much enzymes or proteins that don't function right now. They need to be cleaved or something needs to be modified in order for them to work. So what happens is that angiotensin, when it senses renin, so renin is going to act in a manner to convert angiotensinogen into angiotensin 1. And what happens next is angiotensin 1, goes through our circulatory system. It comes in it goes near our lungs, and our lungs have what are referred to as angiotensin converting enzymes. So let me just write this here. Angiotensin converting enzymes, or ACE. Do not confuse ACE with ACHE, which is acetylcholine esterase, which breaks down acetylcholine when it's used as a neurotransmitter at our neuromuscular junctions. So this angiotensin converting enzyme converts angiotensin 1 into angiotensin 2. And angiotensin 2 acts in a way where aldosterone is released. And we know aldosterone acts on our distal convoluted tubule to promote water to come into our circulatory system. So notice that the cells released renin when there was low blood pressure. So if you retain the water, it's going to act in a manner to increase our blood pressure. So that is a secondary role that you should know the kidney plays, controlling blood pressure. Another one is Osmond Regulation, and we already talked about that in terms of ADH, how ADH can bring the osmolarity of our blood down. One of the last roles that our kidney plays is in terms of acid-base balance. So when our blood gets too acidic or basic, it serves a role to counterbalance that. So if we go back to the original Bohr equation, so the Bohr effect, CO2 goes to H2O. Typing. H2CO3, which goes to H plus and HCO minus 3. Now, We know the kidney can absorb and secrete. So let's view our reasoning on those terms. So what would happen if our blood got too acidic? So blood got too acidic. Well, if we're too acidic, we know we have a high concentration of hydrogen ions. So we want to lower that. So what our kidney's response is going to be is that we are going to absorb, or secrete H+. Notice that when we were talking about secrete, it typically means to get rid of, because we're going to put it in our kidney, which is going to be eventually eliminated. So we're going to secrete hydrogen ions, and we are going to absorb minus or hydroxide ions. And if our blood gets too basic, that means we are increasing our hydroxide ion, or decreasing our hydrogen ions. We're going to act in a manner that's we're going to reabsorb hydrogen ions. And we secrete our excess hydroxide. Just one last thing that I didn't really talk about was, just right here. So we know this was our, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and our collecting duct. So I said I'd come back to this and talk about why. Why is water moved out and then solutes? H2O, we'll just say NaCl. Well, why is because we will have, let's say, our blood vessels going here. So notice that the flow of our material is to the right when it comes to kidneys, and then our circulatory system, we'll say, is to the other way. So what actually happens is the solutes that are first released, let's say NaCl, chlorine, or sodium, these are going to come in and go into our circulatory system. And what happens then is if we move solutes, we increase our osmotic pressure, and water goes to the side with increased osmotic pressure. So then it will just pick up this water. Notice that we need this to happen. If this did not happen, there will be no driving force for the water or the solutes to come out of the descending or ascending loop of Henle. This is referred to as a countercurrent multiplier. So it's just giving the kidney a driving force to release water or release solutes. Because if we don't remove this water or the solutes, they will eventually, the law of mass action, they will eventually become saturated and actually provide a force to go back into the kidney, which is really harmful.